Hello Biology 367 students, and welcome back to this video lecture series on molecular biology. In this lecture, we're going to start Chapter 3, covering an introduction to gene function. In this lecture, we're going to go into an overview of gene expression, starting with DNA transcription into RNA and RNA translation into protein. We're going to talk about DNA replication, and then we're going to go into a bit of an overview of transcription. Sections covered in this lecture are in Chapter 3, 3.1, 3.2, and in Chapter 20, 20.1, and 20.2. We come back to the central dogma, which describes the flow of information from nucleic acids, DNA, and RNA to protein. Producing a protein from DNA involves both transcription and translation. Transcription occurs when the DNA message is copied into RNA. It is called transcription because the code is copied in the same language, that is, a sequence of bases. Translation, on the other hand, involves the conversion of a sequence of bases on RNA into an amino acid sequence on protein. In transcription, one of the strands of double-stranded DNA is used as the template to synthesize a complementary strand of RNA. This is called the template strand, also known as the antisense strand. The other strand of DNA is known as the non-template strand, or the sense strand, because it is in the same sense as the transcribed RNA. A codon is the 3-base or nucleotide sequence that determines what amino acid is used in the translated protein. Chapter 3 gives an overview of the entire process of gene expression from DNA to protein. Before we get into that, though, in this lecture we'll focus first on DNA replication. In principle, we can imagine a few different ways in which a DNA molecule can be replicated. In semi-conservative replication, one double-stranded parent DNA produces two double-stranded daughters, wherein each daughter has one parental strand and one newly synthesized strand. Another possibility would be conservative replication, wherein the replication of one parental double-stranded DNA would produce a new double-stranded DNA with two newly synthesized daughter strands, while the original double-stranded DNA keeps both of the original strands. A third possibility would be dispersive replication, wherein duplication of a double-stranded DNA molecule results in two daughter DNA molecules that both have elements of parental and newly synthesized DNA in each strand. In order to tell which of these three hypothetical scenarios occurred in nature, Meselson and Stahl carried out an experiment in 1958 using isotopically labeled DNA. In these experiments, they labeled DNA with nitrogen-15. Nitrogen has two stable isotopes, nitrogen-14, which makes up 99.6% of the element, and nitrogen-15, which naturally makes up only 0.4% of the element. It is non-radioactive, but nitrogen-15 is more massive than naturally abundant nitrogen-14. Based on density, DNA labeled with enriched nitrogen-15 can be distinguished from DNA which is naturally abundant in nitrogen-14. In their experiment, Meselson and Stahl first grew E. coli in a growth medium that was enriched in nitrogen-15, and this heavy isotope was incorporated into DNA. They then switched the main nitrogen source to the naturally abundant nitrogen-14, and the dividing cells replicated DNA with the light isotope. In the conservative DNA replication hypothesis, this would result in new double-stranded DNA that incorporates the light isotopes in both strands. The original heavy DNA and the new light DNA could be separated by density gradient ultracentrifugation, which I will explain in a minute. In the semi-conservative hypothesis, DNA replication would result in two double-stranded DNA molecules generated from the original heavy DNA, each with the same density, having one heavy strand and one light strand. If these divide another time, each of these two DNA molecules will produce one double-stranded DNA with a heavy strand and a light strand, and another DNA molecule with two light strands. These can all be distinguished by density gradient ultracentrifugation. In the dispersive DNA replication hypothesis, the heavy DNA will be diluted in each strand of double-stranded progeny, and the replicated DNA will become uniformly less dense with each generation. Again, as I said, DNA species of different density can be separated by density gradient ultracentrifugation, which uses a solution of cesium chloride spun in an ultra-high-speed centrifuge. Cesium chloride is a salt that, when dissolved in water, dissociates into constituent ions, a positive cesium ion with an atomic mass of 133 and a negative chloride ion with an atomic mass of 35. Because of the relatively high mass of cesium, when the solution is spun in an ultra-high-speed centrifuge, the ions experience a strong force towards the bottom of the tube, which is opposed by diffusive forces in the solution, which tend towards distributing the ions throughout the tube. Eventually, an equilibrium is reached, and the ions settle into a gradient with a higher concentration near the bottom of the tube. Because of the high mass of cesium, the density of the solution is higher at the more concentrated end of the gradient. When DNA is also dissolved in this solution, it will settle to a position in the tube where the density of the solution is the same as the density of the DNA. Because DNA absorbs at a specific wavelength, its position can be determined by shining UV light through the tube, and in this way, Meselson and Stahl distinguished DNA species of different density. In laboratory conditions, E. coli doubles every 20 to 30 minutes, so by extracting DNA samples from the cultures at these intervals, they could measure the density of DNA species in successive generations. At different time points, they were able to see distinct populations of DNA with different density. These results were consistent with the semi-conservative replication hypothesis. At generation 0, before any replication of DNA with light nitrogen, there is a single band corresponding to DNA with 2 heavy strands. At generation 1, after one doubling, there is a single band at a less dense position corresponding to DNA with 1 heavy strand and 1 light strand. At generation 2, after a second doubling, there are 2 distinct bands at different densities, one corresponding to DNA with 1 heavy strand and 1 light strand, and one to DNA with 2 light strands. Going back to the expected results of the different hypotheses detailed in the previous slide, these experimental results supported the semi-conservative hypothesis, and thus Meselson and Stahl concluded that DNA is replicated semi-conservatively. So we now have a picture of semi-conservative replication of DNA. Before being copied, double-stranded DNA must unwind into single strands, which each template the synthesis of a new daughter strand. When the strands come apart, a replication fork is formed, where DNA synthesis occurs. During replication, DNA strands separate at a defined position in the sequence, called an origin of replication, making a replication bubble. At the end of the bubble is a replication fork. The replication fork moves outward as the bubble grows and complementary DNA is simultaneously synthesized by DNA polymerase. Once again, DNA is synthesized from 5' to 3'. I explained this last lecture, but I will say it again here because it is important. DNA polymerase can only add a nucleotide to the 3' end of a growing strand. Furthermore, DNA polymerase can only elongate a strand. It cannot make a new strand from scratch. As the replication fork moves outward from the origin of replication, anti-parallel parent strands are further separated. Simultaneously, DNA synthesis occurs as the fork moves outward. DNA polymerase can only synthesize DNA from 5' to 3', and the strand that is being synthesized in this direction towards the replication fork continues as the template strands are separated and the fork moves outwards. The other daughter strand is synthesized from 5' to 3' away from the replication fork, and as the replication bubble grows and the fork moves outwards, this leaves a gap in the newly synthesized DNA on this strand. Somehow this gap needs to be filled. In the 1960s, the husband and wife team of Reiji and Tsuneko Okazaki showed that DNA is synthesized on one strand, called the leading strand, continuously, while the other strand, called the lagging strand, is synthesized discontinuously. This is termed semi-discontinuous replication, with the daughter strand that is being synthesized from 5' to 3' in the direction towards the replication fork, growing continuously, and the other daughter strand, synthesized from 5' to 3' away from the fork, being synthesized discontinuously in multiple fragments. The Okazaki team performed several experiments with radioisotopically labeled DNA to demonstrate this. I won't go into details here, but the textbook explains this in section 20.1. The discontinuous pieces of DNA making up the lagging strand are thus called Okazaki fragments. These fragments are subsequently linked by DNA ligase. We know that DNA polymerase can only synthesize DNA from 5' to 3', and that necessitates semi-discontinuous replication of double-stranded DNA. Furthermore, DNA polymerase can only add nucleotides to an existing strand. That is, it needs a primer to polymerize DNA. In 1985, Tsuneko Okazaki established that in vivo, DNA replication was primed by RNA primers. These RNA primers are short 10 to 12 nucleotide sequences that are synthesized by an enzyme called primase. These stretches of RNA need to be removed and replaced with DNA to complete replication. In E. coli, DNA polymerase I is an enzyme that degrades the RNA sequence of a downstream Okazaki fragment, replacing it with newly synthesized DNA that is added to the upstream Okazaki fragment. DNA polymerase I has two enzymatic activities. It has a 5' to 3' exonuclease activity that removes the RNA nucleotides from the 5' end of one Okazaki fragment, and it has a DNA polymerase activity that replaces the digested RNA nucleotides with DNA nucleotides added to the 3' end of another Okazaki fragment. This replaces the RNA, but it still leaves a single-stranded nick in the daughter strand. This nick is repaired by DNA ligase. So far, we've talked about DNA synthesis at a replication fork at one end of a replication bubble. But in the next slides, we'll see that this can occur at replication forks at both ends of the replication bubble. This is called bidirectional replication. There are also examples in nature of unidirectional replication, rolling circle replication being one example in your textbook. I won't discuss that here, however. Instead, we will focus on bidirectional replication in this part of the lecture. In the 1970s, researchers Gurasitz and Wake showed that DNA replication is bidirectional in the bacterium Bacillus subtilis. They fed the bacteria thymidine radioisotopically labeled with tritium, a radioactive isotope of hydrogen. Labeled thymidine is incorporated into DNA. They determined the direction of DNA synthesis by first feeding the bacteria with thymidine containing a low proportion of the radioactive label, and then fed the bacteria with thymidine containing a high proportion of radioactivity. They produced images of replicating DNA based on exposure to radioactivity, like the one shown here. The faint lines show DNA that was synthesized with labeled thymidine during the low radioactivity pulse, and the darker lines show DNA synthesized during the high radioactivity pulse. We see dark lines at both ends of a replication bubble, indicating that the DNA replication is bidirectional. Around the same time, Huberman and Tsai did similar experiments in eukaryotes and showed that in the fruit fly Drosophila melanogaster, DNA replication is also bidirectional, except in these studies, they swapped the order of the high and low radioactivity pulses. In bacteria which have circular genomes, bidirectional replication of circular DNA resembles the Greek letter theta. DNA replication begins at the origin of replication. Again, this is a defined sequence of DNA where replication machinery assembles and replication is initiated. The length of DNA that is replicated following one initiation event at a single origin of replication is called a replicon. E. coli has a 4.6 million base pair genome on a single circular chromosome with a single origin of replication. Therefore, it has one replicon. Most prokaryotes have chromosomes consisting of one replicon. On the other hand, eukaryotic chromosomes typically have multiple origins of replication, and therefore multiple replicons. I will pause here, but to summarize what we've discussed so far, I've shown you that DNA replication is semi-conservative and semi-discontinuous, and we've seen that DNA synthesis occurs bidirectionally from an origin of replication. In the next part of this lecture, we'll focus in more detail on the enzymes involved in DNA replication.